The FDA has expanded approval for Servius Tibsovo to include its use in patients with relapsed or refractory myelodysplastic syndromes carrying a susceptible IDH1 mutation, making it the first targeted therapy for this specific group. Tibsovo, already approved for treating acute myeloid leukemia and cholangiocarcinoma, targets and inhibits mutated IDH1 enzymes. The FDA also approved Abbott Laboratories' real-time IDH1 assay as a companion diagnostic. Tibsovo's safety profile aligns with previous findings, and its label includes a boxed warning for differentiation syndromes in MDS and AML. An international team led by ETH Zurich has conducted the first study on the Rocky River, a tributary of the Congo, known for its exceptionally dark water. The river's unique darkness is attributed to low sediment content and high levels of dissolved organic substances, creating what researchers refer to as jungle tea. Despite its vast, pristine rainforest watershed, the Rocky had never been scientifically studied. The research revealed it to be one of the richest blackwater river systems globally in terms of dissolved organic carbon compounds, surpassing even the Congo River and the Rio Negro in the Amazon. The study sheds light on the carbon cycle of the Congo Basin and underscores the ecological importance of this mysterious river. Researchers from the Fraunhofer Institute for Chemical Technology have conducted a large-scale study to investigate the efficacy and environmental impact of biodegradable bags, known as biobags, for collecting organic household waste. The study aimed to analyze the decomposition behavior of biobags in existing organic waste treatment plants, assess potential environmental effects, and determine public interest in adopting biobags. The research focused on whether biobags could serve as a viable alternative for collecting household organic waste, considering the prevalent issue of plastic contamination in organic waste processing facilities. The study involved distributing various types of bags to tens of thousands of households and examining the resulting compost for the presence of plastic fragments. The findings aim to provide valuable insights into the practicality of using biobags in waste collection and their impact on the environment. Eisai and Biogen have presented data on a subcutaneous version of Alzheimer's drug Leqembi, aiming to address concerns about healthcare infrastructure burden. The injected Leqembi demonstrated comparable efficacy to the intravenous version in removing the toxic amyloid protein associated with Alzheimer's, according to an extension of the Phase 3 Clarity AD trial. Despite positive efficacy results, the subcutaneous version showed a numerically higher rate of the amyloid-related imaging abnormalities compared to the intravenous version, raising safety concerns. However, analysts argue that the difference was drawn from a smaller number of patients, and the overall profiles of these abnormalities were deemed more similar than dissimilar. Eisai and Biogen are considering the subcutaneous version as a weekly self-administered injection, potentially offering a more convenient dosing option. The FDA has granted approval to Eli Lilly's mirikizumab, now branded as UNWO, for the treatment of adults with moderately to severely active ulcerative colitis, making Lilly's first entry into the inflammatory bowel disease market. The drug is a humanized monoclonal antibody that targets the IL-23 cytokine and blocks the IL-23 pathway, which play a central role in various autoimmune disorders. UNWO is the only FDA-approved ulcerative colitis treatment that selectively targets the P19 subunit of IL-23. The FDA has approved Santhera Pharmaceuticals' corticosteroid vamorolone, now branded as Agamree, for the treatment of Duchenne muscular dystrophy, or DMD. The drug, an oral suspension indicated for DMD patients aged 2 years and older, has transformational potential and could reshape the DMD treatment paradigm. Agamree works through a unique mode of action, eliciting differential effects on glucocorticoid and mineralocorticoid receptors, allowing for better tolerated side effects compared to standard corticosteroids. Catalyst accepts to launch the product in Q1 2024. Genentech's Vabysmo has received FDA approval for treating macular edema following retinal vein occlusion, a condition impacting over 1 million people in the US. The drug demonstrated effectiveness in improving visual acuity for its patients. Vabysmo, already approved for wet age-related macular degeneration and diabetic macular edema, targets angiopoietin 2 and VEGF-A pathways. Genentech plans to launch a prefilled syringe in 2024 for easier administration and is exploring additional indication for Vabysmo through upcoming studies. The drug is well tolerated with conjunctival hemorrhage as the most common adverse reaction. A University of Tübingen study reveals unique variations in the immune response within nasal passages, emphasizing individual differences in the secretion of immunoglobulin A. The research published in Microbiome suggests diverse reactions to microbes, potentially explaining varying infection susceptibilities. Examining the secretion of these immunoglobulins' interaction with Staphylococcus aureus, including the antibiotic-resistant MRSA variants, the study hints at therapeutic possibilities for manipulating these interactions. Analysis of nasal swabs from 50 individuals indicates a direct link between higher secretion of immunoglobulin A levels and lower bacterial colonization, suggesting a role in regulating microbial populations. The findings offer insights into the collaboration between the immune system and the microbiome, with potential implications for future treatments, though further research and investment are needed for translation. Researchers at the University of Heidelberg's Biochemistry Center have examined the role of a specific membrane proteins in the mobility of sea urchin sperm, providing insights that could lead to novel contraception methods. During the maturation of sperm cells, known as capacitation, their mobility must be increased for optimal functionality. Membrane transporters responsible for transporting ions into the cell play a crucial role in this final maturation process. Using cryo-electron microscopy, the scientists decoded the molecular structure of a key membrane transporter for sperm, revealing its functional units and their interactions. These findings paved the way for potential substances that influence this mechanism, potentially activating or deactivating protein functions. While further research is needed to understand the transferability to human sperm, the study holds promise for developing new approaches to treating infertility or intentionally preventing fertilization for contraception. Researchers from Baylor College of Medicine have discovered a small molecule called 5D4, a top BP1 inhibitor, could effectively inhibit the growth of breast and ovarian tumors in mouse models. The top BP1 gene is associated with the challenging to drug MYC gene, known for its involvement in various cancers. By targeting top BP1, the researchers found that the drug enhances the activity of another gene, MYZ1, which in turn limits MYC's cancer-promoting abilities. The drug worked even more effectively when combined with the PARP1/2 inhibitor talazoparib. The researchers plan to further develop 5D4 for human use, optimizing its anti-tumor effect in combination with other drugs and minimizing potential toxicities. The study offers promising insights into new therapeutic strategies for breast and ovarian cancer. Eli Lilly's Zepbound has received FDA approval for chronic weight management in adults with obesity or excessive weight and related conditions. Zepbound, containing tirzepatide, is the first treatment activating two key hormone receptors linked to excess weight. The FDA warns against combining it with Mounjaro or similar medications. Approval is based on positive results from phase 3 trials showing statistically significant weight reduction. With this, Lilly enters a lucrative market alongside Novo Nordisk Wegovy, and analysts predict the obesity drug market could hit $100 billion by 2030. Zepbound is said to be available in the US by year-end, priced around 20% lower than competing options, and regulatory reviews are underway for global markets. 4D Molecular Therapeutics aerosolized gene therapy 4710 has shown promising results for cystic fibrosis patients with high protein expression, stable improved lung function, and enhanced quality of life over 12 months. Airway biopsies indicated robust CFTR protein and RNA expression. FDA feedback on development plans is expected in Q1 2024. The University of Altenberg and Bremen's joint research reveals that the impact of cold on the nutrient content of kale depends on the variety. The study presented at the North American Cystic Fibrosis Conference explores how genetic differences in temperature affect the levels of flavor-enhancing compounds in kale. Known for its high content of secondary plant compounds, kale, including the responsible glucosinolates for its distinctive taste, shows a varied composition influenced by both environmental factors, especially temperature, and genetic factors. The research team found that cold increases glucosinolate concentration in some kale varieties while decreasing it in others. The study suggests that considering both temperature and cultivated variety is crucial for obtaining plants with an improved nutrient content. Researchers at New York University have identified a small molecule, CBD3063, that effectively alleviates 4 types of acute and chronic pain in rodents without the side effects associated with the widely used non-opioid painkiller gabapentin. CBD3063 alters the calcium channel on pain-sensing neurons, reducing neurotransmitter release and pain. In experiments, CBD3063 worked at a lower dose than gabapentin, showing comparable pain relief. The researchers see potential for CBD3063, but acknowledge the need for more experiments to understand its long-term effects and potential tolerance issues before progressing to clinical trials. Bristol Myers Squibb has gained FDA approval for Augtyro or repotrectinib, a tyrosine kinase inhibitor, to treat locally advanced or metastatic ROS1-positive non-small cell lung cancer. The approval is based on positive results from the Trident-1 study showing efficacy in both TKI-naive and TKI-penetrated patients. Augtyro aims to address treatment resistance in ROS1-positive NSCLC. Charles River Laboratories and Asia have partnered to accelerate drug development programs targeting neurodegenerative diseases and cancer. Asia will utilize Charles River Logical Platform, an AI-powered drug solution for its drug candidates focused on Alzheimer's, Parkinson's, Huntington's diseases, prostate cancer, and multiple myeloma. Asia, specializing in causal AI and digital twins, aims to unravel the genetic and molecular complexities of neurodegenerative disorders to advance the discovery of treatments. The collaboration is expected to provide valuable insights for rapidly translating discoveries into novel drug candidates. The FDA has approved Let's Get Check's Simple 2 test, the first at-home test for chlamydia and gonorrhea in the U.S. Let's Get Checked, known for remote testing during the pandemic, offers an over-the-counter solution allowing adults to collect samples at home and mail them to a lab for results. The test, expected to provide results within 2 to 5 days, includes a prepaid shipping label for sample return. Within 1.6 million chlamydia and over 700,000 gonorrhea cases reported in the U.S. in 2021, the FDA deems this clearance a public health milestone, addressing the rising cases of these common STDs. Pfizer and Astellas have gained FDA approval to broaden the use of their prostate cancer drug Xtandi for non-metastatic, castration-sensitive prostate cancer with high-risk biochemical recurrence. Xtandi is the first and only androgen receptor signaling inhibitor approved for this indication, with or without gonadotropin-releasing hormone analog therapy. The approval, accelerated through FDA programs, follows Xtandi's success in reducing metastasis or death risk by 58% in the EMBARK trial. The UK has granted regulatory approval for Casgevy, a CRISPR gene-edited medicine developed by Vertex Pharmaceuticals and CRISPR Therapeutics, making it the first CRISPR-based therapy authorized for use globally. Casgevy targets blood diseases, specifically sickle cell and beta thalassemia, by modifying a patient's own stem cells in the laboratory using CRISPR technology. The edited cells are then reinfused, producing functional hemoglobin, alleviating symptoms and potentially offering a lifelong benefit. The approval is limited to patients unable to receive a stem cell transplant from a matched donor, and clinical trials have shown promising results in eliminating pain crises and reducing the need for chronic blood transfusions. Researchers from ETH Zurich and a US startup have developed a laser scanning technology enabling the 3D printing of plastics with superior elasticity. Unlike traditional fast-curing plastics, this advancement includes slow-curing plastics with enhanced elastic properties, durability, and robustness. The technology allows the creation of intricate, resilient robots using various high-quality materials in a single printing process. With the ability to combine soft, elastic, and rigid materials, it facilitates the production of parts with hollow spaces and intricate structures. The process involves real-time scanning of each layer for irregularities, adjusting material quantities for subsequent layers. This breakthrough holds significant promise for advancing soft robotics and crafting sophisticated, resilient structures. Researchers from the University of Heidelberg and the Francis Crick Institute in London have unraveled genetic programs governing the development of gender-specific traits in key mammalian organs. Studying humans, mice, rats, rabbits, and opossums, the team decoded these programs, shedding light on the evolution of gender-specific organ traits. The findings offer essential insights for gender-specific medicine, particularly in organs like the liver, where gender-related differences impact drug processing. The study highlights the swift evolution of gender-specific features activated by male or female hormones during late-stage organ development across mammals. Researchers from ETH Zurich have explored the idea of protogenes as a precursor genetic code system before the emergence of complex molecules on early Earth. They focused on amyloids, protein-like aggregates demonstrating their ability to form, replicate, and bind to genetic molecules like RNA and DNA. The interactions influenced by electrostatic attraction and the sequence of genetic building blocks suggest a potential precursor to universal genetic code. The study highlights the mutual stability gained from the binding, indicating a potential advantage in the dilute conditions of the early Earth. This suggests that molecular cooperation rather than competition may have played a crucial role in the emergence of life. Thanks for watching my Biotech News Update. If you found this video informative, please don't forget to hit the like and subscribe button for more updates. Additionally, all the sources used in this video can be found in the description below. Stay tuned for more exciting news in biotech.